fifth section of the book goes into, remember we're still on adaptive, right? And starts talking about humoral response versus a cell-mediated response. So I mentioned this earlier though, humoral meaning human, right? So a lot of scientists nowadays actually refer to this as just antibody response, because that's what the humoral response is. But I still leave humoral, probably should put antibody in parentheses. You unfortunately need to know both are synonymous. This was probably the easiest response to study when scientists first started studying the immune response, because anybody would know what type of molecule antibodies are? Protein. Remember, that's like if you're not sure, that's the answer, right? Because proteins are the workhorses of our cells, but some of them even work pretty awesome outside of our cells. I have to say, if I had to pick my favorite protein, antibodies rank really high on the list, because they have really impressive abilities that we're going to talk about towards the end of today's lecture. But they have limitations to them, right? Something we'll also talk about. A cell-mediated response, as the name implies, it's cells that are doing the work as opposed to antibodies. And you can also think of it as a response to something getting inside of your cells. Because remember, some pathogens have to get inside your cells to replicate. Who are those? Viruses, right? Some bacteria, too, are intercellular. Some protozoa are intracellular. They got to get inside of our cells. So we have to have a way of detecting that something got inside, right, and destroy that invader that's inside the cell. So that's really the job of your cell-mediated response. Your humoral response is, like I said, often just referred as an antibody response. And one of the limitations to antibodies is they don't go inside of our cells, right? They can bind to the outside of a cell and target it for destruction, but they do not go physically into our cells, right? So that leaves that area vulnerable. But they are pretty awesome outside of our cells, right? And have a lot of ability outside of our cells. So this is from that page, right, which unfortunately the internet is no longer hosting, but I found my copy. So one of the things that y'all notice, I went on a grading spree yesterday. If for some reason you haven't done an assignment, do it before I go on my next grading spree so you don't lose points, right? And the comment next to it probably says such if you click on your grades. So this one was really good, right? Killer T cell. We'll literally, and notice what I like about this is they're inside the cell, right? So this is a virus-infected cell. This is a bacteria inside of a cell. This is a cancer cell. So this would be a killer T cell, or what do we typically call them? Cytotoxic T cells. And they have which CD marker on them? 8. 8. Remember, sounds like eat, because they kill, they eat. Okay? And then this is a type of cell. It's a differentiated lymphocyte. And I like this picture, right? Because they literally secrete the antibodies, so they're like, he's like spitting them out of his mouth. And they're binding to the bacteria, which the look on their faces says it all, right? This is not a good thing, that antibodies are binding to them. The one thing I don't like about is how the antibody, they show binding. But we'll talk about that later. We'll do better. So does anybody know what this differentiated from? It's called a plasma cell, but it was something before it turned into a plasma cell. It's a B cell, right? Before that, it was a B cell, right? Which actually had this antibody on its surface as its receptor for the antigen. But at this point, it's differentiated into a plasma cell, something we're going to talk about today, and secreting antibodies, which are proteins, right? And we'll go into the different types of antibodies. There's actually 5 different types. We're going to go into the 4 major types today. So, as I said, humoral sometimes also referred to as antibody. See, at some point I knew I put it in percentages. And so, as I said, good against extracellular antigens, stuff outside of our cells. Okay? So those bacteria are outside the cells that the antibodies are binding to. Where the cell-mediated is, this is our good response against stuff that gets inside of our cells. So that's the two broad responses, two choices. So how do they decide which way they're going to go? And remember, cytokines are those chemical messages that our immune cells use, right? And who's the big talkers? The helper T cells. So a lot of times you'll see them written T with a subscript H for a helper T cell. These are the ones that are going to talk, right? Release those cytokines that are going to talk to activating B cells or cytotoxic T cells. But as I said, right, B cells won't directly help kill a viral infected cell. The antibody itself cannot cause destruction of that virus inside the cell. It can help us detect it, right, but it will not destroy that cell. It needs help from the other cells of the immune system to achieve that goal. Cytotoxic T cells, on our other hand, won't directly kill bacterial cells. So say you have a bacterial cell outside of one of your cells. A cytotoxic T cell cannot recognize that bacteria as being foreign and will not kill it. Cytotoxic T cells pretty much only kill our own cells. Because remember, T cells have to have antigen presented to them, right? And only our cells will do that via MHC. If it's for cytotoxic T cells, it's MHC class what? 1, right? And cytotoxic T cells are CD8, right? That matchup you got to remember. We'll repeat it over and over. So this, the details of this, right, which I'm going to skip over a lot of. As you can see, I put nice bold, not on the exam. For T cells, how do we decide which way we're going to go, or how much in one direction or the other we're going to go? There are two subsets of T cells. There's subset 1 and subset 2, hence the 1 and the 2. What's the difference? They're going to release different sets of cytokines to direct which way we're going, right? Are we going antibody humoral response? Are we going cellular response? Are we going to do both, right? And for some diseases, like the study of these two subsets and what happens is really important in understanding on why, for some people, right, their body chooses to go the wrong way. So think about this. If you have an intercellular pathogen, what type of response do you want? If a bacteria has gotten inside your cell, you want a cell-mediated response. You want cytotoxic T cells recognizing those bacterial infected cells, and you want to kill those cells, right? For leprosy, that's an intracellular bacteria. Some people just produce antibodies. Doesn't do you any good because the antibodies can't get inside the cells. Only when that, for the short period of time that cell is outside of the cells, it may help. But it's not going to help clear that infection, right? The good news is it's a bacteria, right? So we can take antibiotics and kill it nowadays, right? Not in the, they've been talking about leprosy since biblical times, right? People still get this disease, right? It's treated on outpatient basis, and we don't see quite the problems that we used to. But again, different people respond differently. Why would one person respond one way and one person respond a different way? Your immune system acts differently, but where does that information come from? It's encoded in your DNA, right? And so leprosy tends to run in families. Why? Because your DNA is the same, right? So this defect of not effectively mounting a cellular response instead of humoral antibody response is inherited. And then families usually tend to live in the same areas, right? you're exposed to the same pathogens, right? That's why you see the trends, right, that you do with certain diseases, right, among families, right? It's genetics, right? It all comes down to DNA. And then, of course, environment, as we know, is a big factor as well. So yeah, different cytokine profiles by these different subsets, right? So IL, remember, stands for what? Interleukin. And interleukin-2 is really important for proliferation of cells. It's one of the few, right? TNF stands for tumor necrosis factor, right? Another important. IFN, interferon, right? So notice that, like, these are things that are very common with cell-mediated response, right? We're getting a different profile, right? Different talking between white blood cells, interleukins between white blood cells, when you're getting that antibody response, which is a Th2 response, right? And there are feedback loops, right? So notice again, talking, differentiating, directing which path they're going to go, okay? But this diagram, thankfully, we're not going to go over. We're going to skip it. Right? But one of the important parts is, how does this whole thing start, right? Mainly dendritic cells, right? And notice other cells get involved with the antibody mediated. So remember these guys, the toll-like receptors? And again, it's all about DNA, y'all, because look, that signal gets sent internally that says, okay, we need inflammatory response, we've got bacteria, right? Bacteria binding to the receptors on the cells, or stuff is getting inside the cells, internal bacteria, or viruses. Especially viruses, we want the interferon response, right? Because remember, interferon is going to bind to neighboring cells and warn them that viruses are on board, and they get ready to destroy that virus if it comes in. And they're going to sacrifice themselves in that process. So, really important, right, to the start of the whole thing is that alarm system, that recognition, right? Those patterns that we recognize, like lipopolysaccharide and flagellin, and that's supposed to be double-stranded RNA. I just noticed a typo. It's supposed to be DS. But you know, viral DNA, you could even write there, right? We have these receptors, as you saw in the previous picture, on the membranes, right? Toll-like receptors, pattern recognition receptors, inside endosomes within the cells, right? Cells that are injured, things that happened, right? They're going to communicate. IL stands for interleukins, right? INF is interferon, and TNF, right, are one of the few acronyms I do expect you to know what they stand for, right? Because it's going to help you out. What you don't have to know this semester is Th1 versus Th2, which is which. But know, right, that we do make a choice, right? And sometimes we go too far in one direction or the other, and that's inappropriate for the response, right? And, of course, there is communication, right? Feedback. All right, so sometimes, as I gave the example of leprosy, immune responses can damage cell structures if they're too strongly stimulated. So meningitis, itis just refers to inflammation, right? The meninges are the membranes that surround your brain and your spinal cord. So excessive pressure on nerves, paralysis, and even death, if we have pathogens that get into those areas, and sometimes the immune response can be just as damaging as the pathogen. With gonorrhea, because it can damage the ciliated cells associated with the female reproductive tract that help sweep the egg down the fallopian tubes into the uterus, those get damaged by gonorrhea infection and can lead to sterility, right? Because the eggs no longer are being directed to where they need to go. There are some pathogens out there that produce what we call super antigens. They're not so super in our body. They sometimes activate responses we don't need responding, or they keep the response going even when we don't need it, right? So they usually result in excessive levels of cytokines released from the helper T cells. They kind of get the helper T cells stuck, right? And they just keep talking, and they should really shut up at this point, but they don't. And then we believe that there's some cross-reactivity that sometimes happens where we've produced antibodies to bacteria out there, and unfortunately they bind to things in our body and cause damage to our body, right? One of the things I find most interesting is the Abo Blood Group, right? So remember, O actually meant no antigen, but you have either the A or B antigen, or like me, no antigen on the surface of your red blood cells as it relates to that antigen group, right? That we talk about for blood typing. It really is a sugar protein association on the outside of your red blood cells. Well, usually, as we'll talk about, in order to get your body to make an antibody, you've got to be exposed to that antigen. For the Abo blood group, not the case. Like, I, over time, have been exposed to, unfortunately, probably bacteria that have very similar antigens to the Abo blood group, which for them might have been a good thing to evolve, because sometimes people wouldn't then produce antibodies because you don't want to make antibodies against yourself. That'd be autoimmune disease. You'd be attacking yourself, right? But in this case, most of us have been exposed to bacteria. The good news is the type of response and the type of antibody is not the same as the one you do for RH, right? So you produce what we'll talk about later, IgM. And that antibody has some limitations. One of them, it doesn't cross the placenta to the baby, unlike RH, which does. But there's a reason why you produce IgG, which is an antibody that can cross the placenta against the RH antigen, which when people say, oh, I have, O positive or A negative type bloods, you're actually talking about two separate antigen categories when you give that blood type. You're talking about your status as far as the ABO and RH. And for RH, you either have that antigen, you're either positive or you're negative, right? The bad news for moms is if you're negative, do you have that antigen? No. If you're exposed to that antigen, can you make those antibodies? Yeah. Can those antibodies cross the placenta and hurt your baby? Yes. Can your baby be positive even though you're negative? Yes, because dad, yep, you can blame it on dad, right? He donated the positive DNA as it relates to RH. So when I think about that one as far as cross-reactivity, it helps really help me remember this, right? That there are things out there, unfortunately, that are similar to us, and we'll produce antibodies against them, and that can create problems for us, especially with blood donation, as far as the Abo blood group goes, or other things. Viruses result in immune-based destruction of large numbers of cells, right? So especially with hepatitis B. So hep refers to what organ? Your liver. Itis refers to, again, an inflammation. B is just a group, right? So as we discovered things, right, we gave them a letter designation. I think we're up to A, B, C, D, E, F, G. I think we're up to G for hepatitis, by the way. But you don't hear about most of them, right? You hear about A and B, which we vaccinate against now. We don't have a vaccine against C. And remember, D is just a variation kind of B. We talked about this confusing situation. So vaccine against B takes care of D as well. But there is E, F, and G out there. But they're not common in the United States. Yeah, for what? C. And that problem with C is we don't have a vaccine against it. Yeah, that's the scary thing about that. But again, it's causing destruction to liver cells. Liver is kind of an important organ, right? Does a lot of really cool stuff for us. So that's the problem, especially with hepatitis B and even with C, right? And the good news is they've got some drugs now, right, that can really help out with C, especially since we don't have a vaccine. But yeah, as we've talked about, our body will kill virus-infected cells. Well, you know, if you can't get the virus, if you can't stop that replication cycle, right, you create a lot of damage in the process, right? Sometimes we've got plenty of cells to spare, in some places not so much. And the liver is really not a place where you want to, you know, go on a mass shooting spree, killing a whole bunch of your own cells, right? That organ is important. It's probably one of the largest if you don't consider the skin. But your skin is the largest organ in your body. But liver's pretty big, pretty important. So we started out with the features of adaptive. We talked about T cells. We talked about antigen presenting, antigen processing, and then antigen presenting cells, and even the professionals as it relates to that. Right now, we just briefly mentioned that we do have choices, right? Antibody versus cell mediated. And sometimes we'll do both, right? And that's good, right? Sometimes we'll have a balance. Sometimes we teeter-totter, unfortunately, the wrong side, depending on what came in. And this really boils down to a lot of times a person's genetics, right? Which is obviously not easy to change. But we can augment these things nowadays with drugs, right? And different types of treatments. So now we're really going to focus on B cells and their main job, which is the production of antibodies, which are proteins, right? And these leave the cells, and so these are really good for our extracellular response. So section 6 of your book, in this chapter, make sure I hit A, goes over this. So this is a good diagram, right? Kind of a good flow chart. I remember some of this I'm not going to kind of hold you to. But let's start here in the beginning. Naive Cd4 cell. What's another name for that cell? I didn't cover it up, by the way, in the diagram. That's another name for I just said it. CD4 T cells are called helper T cells. So some antigen presenting cell, right, in this case dendritic cells are the main ones because who are we talking to? A naive T cell, right? And they're not showing the direct interaction there, but helper T cells have MHC what? You have two choices, right? Two. Right? They have two, right? And so they're going to communicate with each other, and it's going to cause the proliferation of that cell if they match up. And so we're going to get a whole bunch of helper T cells, and then memory helper T cells. So in case it comes back, right, an antigen presenting cell, in this case, it doesn't have to be a dendritic cell, it can be a macrophage, can start this process again. But for helper T cells, they have a choice now. Which way do we need to go? And it's really going to depend on this communication, what receptors were triggered in the beginning, as to which way they're going to go, right? So for a cell-mediated response, it's going to be what I call TH1, right? Which one you don't have to worry about. Which is going to stimulate, this is our cytotoxic T cells, right? This is our cell-mediated response, our cytotoxic CD8 T cells. But they've got to be activated too, right? So they're going to get cytokines that are going to help with the activation from helper T cells. But, right, a dendritic cell is going to have to present antigen via MHC class what? To this naive CD8 cytotoxic T cell. 1, right? And again, they're going to proliferate, so we're going to get a whole bunch of them. Notice the little dots inside, because then they're holding, you know, perforin and granzyme. They're going to go and literally dump on cells that are expressing MHC class 1 with this antigen. We're going to produce memory cells, right? And then notice again, macrophages or a dendritic cell, right, can activate a memory cell in case the bad guy comes back again. And we go through the process again. The other choice, right, is helper T cells subset 2, where we get the humoral or antibody response. Who are they going to talk to, right? So they're going to release cytokines. They're going to talk to B cells. And what they don't show right here is the B cell getting activated, because this doesn't always necessarily need to happen. Was one thing I don't love about this diagram. So here is that naive B cell, right? It sometimes does need to be activated, and probably the reason why they didn't show it is it doesn't always. Something we're going to talk about today, right? But it's going to present antigen to the helper T cell that's already been activated up here. You see that? Right, way up here, helper T cell activated. He's been activated. Right, naive B cell becomes activated. I guess they are showing it. So B cells, do you guys remember which MHC they're going to present? Since they're talking to a helper T cell, this is 2. And they're going to differentiate, right? They're going to proliferate. They're going to produce plasma cells. And notice they list 4. These are the 4 majors. There's one more. But these are the 4 major classes. There's 5 of antibodies. So IG, remember, stands for immunoglobulin, right? So class M, G, A, E. And there's D, which we're not going to talk about. And then again, if the bad guys come in, again, the memory B cell can be activated, right? And we go back through the process again of producing more plasma cells that can secrete antibodies. So that's without the things hidden. Okay, so our best understanding of how B cells produce antibodies is in the response to protein antigens from foreign sources. And as you saw in that diagram, usually requires, oh, what happened? T cell, helper T cell help. But the B cell itself has to bind and can bind to that foreign antigen. We'll look at this process. It ingests and processes it and presents it on MHC class 2 to the helper T cell. Right? Cytokines are going to release. The B cell multiplies. It differentiates into antibody secreting cells. We call these plasma cells. We don't call them an activated B cell per se, right? Because they dramatically change. What's the drawback in this? Even with a T cell response, right? Especially with an antibody response too. The very first time you ever see this antigen, right at the top, remember, it's a naive, never saw this antigen before, B cell that's got to get activated. This can take 7 to 14 days. This is why the second you get the flu shot, it doesn't work right away. Right? Your immune system needs anywhere from a week to 2 weeks if it works, to produce those antibodies. And remember, when you're showing signs and symptoms of an infection, whether it be viral or bacterial, there was a period before you showed symptoms that you were infected, right? We call that the incubation period. The period before you know you're sick, hence why you're going around getting everyone else sick and you don't know it. If you are physically showing symptoms, though, you should stay away from others because you are definitely, right, contagious. So sometimes it was called secondary response, but a lot of us really call it memory response now. Because again, if we get a memory cells produced, if it comes back, those are the cells now that are going to be activated as the memory cells. What's the good news about that? There's no waiting. They're already there, right? And there's a lot of them, right? Instead of that few naive cells we were trying to find, this can be a much faster response, right? Larger scale, and the ability to respond to different antigens is still retained. So, say you're injected with the flu vaccine right here, right? 2 weeks later, you're producing a primary response, right? What if you get exposed or injected with a different vaccine, right? And you get exposed to the flu at the same time, right? Because you've seen A before, right, because you got your flu shot, you're going to respond so quickly that you don't even get sick. You don't even know that you inhaled it, right? And at the same time, the immune system can be responding for the first time to B, which you've never seen before. But again, that first response, right, there's going to be a time when we got to get it going. And that takes literally time. That's the one drawback, right, to the adaptive immune response. It takes time, right? You got to find those naive T cells. You got to find those naive B cells that recognize.
You said it's happening all the time.
All the time. All the time. This is just showing 2 antigens. You could be responding to, I don't know, 5, 6 at the same time. When you're first born, you're assaulted, right? And like when we give babies a whole bunch of vaccines, like people can complain. What we give in vaccines is nothing compared to what you're exposed to when you're first born. It's ridiculous. Okay, so how do B cells present? So they have their B cell receptor on their surface, right? And they can bind to things other than proteins, lipids, carbohydrates, right? It can bind to the receptor. It'll be internalized like phagocytosis. They're going to break it up into pieces, and they're going to present it via MHC class 2. If a T cell, helper T cell, right, in this case subset 2, right, recognizes it, it's going to release cytokines, right, help this guy proliferate. So we're going to make a whole bunch of clones of the same of this, and they're going to differentiate some of them into plasma cells. These actually secrete the antibody that they had on their surface that recognize that antigen. Some of them become memory cells, which are going to stick around. The antibody's released, right? So then they can go and like bind to bacteria. How is that helpful? Well, it basically tags them for destruction. Do you guys remember that name for tagging something for destruction? C3b of our complement can do it. C-reactive protein can do it. Mannose binding lectin can do it. Starts with an O. Opsonization, right? Tagging for destruction. So just the antibodies binding is doing just that, saying, hey, this is a bad guy. You need to destroy it. Who's going to actually destroy it, though? A phagocyte. Who's a phagocyte? Be more specific. A macrophage has receptors for these and neutrophils, right? Have receptors for antibodies, right? And C-reactive protein and different things that we're going to kill them. So the B cell receptor, right, may take up non-protein antigens so long as the antigen fragment can be presented via MHC class 2. The reason why they make this distinction is typically MHCs picks up proteins. But what we found with the B cells is they have a way of somehow loading these things that are not proteins into MHC class 2. So in that way we can respond to things other than just proteins, right? Carbohydrates, lipids. Although proteins, as you know, are the most antigenic. So, okay. That's with T cell involvement. What are T-independent antigens? Well, the name tells you it all, right? It tells you that there are some antigens out there. T cell involvement is not needed to get the B cell to produce antibodies. How'd they figure this out? Either in some mice and some humans that don't have a thymus. Because remember, T cells are named after the thymus, right? That's where they go and get their education, right? Where they become CD4 and CD8 T cells, where we make sure that their MHC class 1 or class 2 is working properly, right? Where they get screened and checked out, hopefully. And then they travel to the lymph nodes and the skin-associated lymphoid tissue called SALT, and your mucus-associated lymphoid tissue, or the Pryor's patches in your small intestine. So, but, okay, here we have humans or mice, right? No T cells, no thymus, no mature T cells. And they're still producing antibodies. So, right there, do you need a T cell to get a B cell to make antibodies? No. But how is this done? Typically with antigens, and we'll see a picture of this, that will bind to a lot of the B cell receptors all at once. We call this string response, right? So these are long polymers that are repetitive, right? So the molecule binds to a lot of receptors on the surface. Because of this, though, we tend to only produce the first type of antibody, which is IgM. This one, thankfully, has a lower affinity than other types of antibodies, and as I said, has limitations. What do you think I mean when I say affinity? An antibody does what? Binds, right? Physically binds to the antigens. Affinity means how strong that binding is, right? How attracted are you, right? What is the affinity? This one is low, right? So, not very good binding happens. Stimulation of the B cell is weaker, often not producing memory B cells. So, okay, we produce an antibody response. It's a weak response. We don't even tend to remember the response. Typically, less antibodies are produced than with T-dependent antigens. Which makes sense, right? Because antibodies we see are pretty powerful. We don't want to produce the most powerful antibodies unless we're sure this is the type of response we want to go with, right? But a little bit of a response sometimes is better than no response at all. Okay? So this can happen. This definitely happens, like I said, to the Abo Blood Group. Right? Even if you haven't been exposed to foreign blood, I've never had a blood transfusion, but I can guarantee you I probably have anti-A and anti-B antibodies of this class, IgM. Now, we draw antibodies kind of as this Y stick figure, and notice here, as I said, right, a lot of interaction between a lot of the receptors on the B cell. But when the B cell proliferates, turns into the plasma cell, there's no memory cells produced, or if any, very little. And they're going to secrete the very first antibody they produce, which is IgM. This is actually 5 monomer antibodies put together. So an antibody monomer is just this one single Y, right? This is 5 of them. Do you see that? A pentamer hooked together. So I always tell my students, remember M for mega big. This is the biggest antibody we produce. And because it's so big, it doesn't cross the placenta, right? It typically doesn't leave the blood vessels unless we've got inflammation and the blood vessels become leaky, right? So this one travels throughout the lymph system, through the blood system, right? And unless it's being made on scene, it really isn't in your tissues in high concentrations. So how amazing are your plasma cells? They can secrete over 2,000 antibodies per second once activated, right? They're terminally differentiation, which means once they become a plasma cell, that's it. That's the end of the line for them, right? So a B cell becomes activated, it proliferates, a whole bunch of clones are made, they differentiate, right? Some of them become plasma cells, some of them are going to become memory B cells. Those plasma cells, right, after their job is done, which can be as little as 2 days, go through programmed cell death, which we call apoptosis. What's important about apoptosis? It doesn't elicit what? Inflammation, right? Because we're like, yep, your lifespan's up, right? But the antibodies they secrete can last for weeks in the blood, right? Even a month, right? They can stay around for a long time. So even though the B cell who made the antibody, he's done, his lifespan is ended, the antibodies themselves can stay around for quite some time. So why is it called a plasma cell? This picture really helps see that. So here's a plain old B cell, right? When it differentiates into the plasma cell, you see all these lines? That's endoplasmic reticulum, specifically rough. And why do we call it rough? What is it studded with? Ribosomes. What do ribosomes make? Proteins. What are antibodies? Proteins that are secreted. The rough endoplasmic reticulum and the Golgi apparatus, right, which is hard to distinguish from the rough endoplasmic reticulum because it gets so huge in these cells, right, or help what direct proteins that are secreted. So that's where they got their name, like their plasma, right, their cytoplasm gets enormous, right, where remember B cells, lymphocytes themselves, when you look at them under the microscope in typical normal blood, right, they've got a big nucleus and little tiny light blue cytoplasm, right? If you were to look at a plasma cell, right, huge cytoplasm in comparison. So antibodies produced by plasma cells. All antibodies from one plasma cell, right, so one of them are identical and will only bind to a single epitope on an antigen. So as you see, we'll zoom in on this picture in a minute. However, recall that a single antigen may have many epitopes, so many different plasma cells may be formed, each with a different specificity. So, okay, imagine this is an antigen, right? Could be a protein, could be a bacterial cell, could be anything foreign, right? As little as, say, 10 amino acids sticking out right here. B cell clone line number 1 made this antibody, right? Only this specific antibody that only binds to this specific epitope on the antigen. Number 5 was made by the 5th clone. It's a completely separate B cell line that was activated. That B cell line only makes this specific antibody. that binds to this specific receptor on this antigen. What's the good news here? Is look, our body in response to this antigen produced 5 different antibodies. It activated 5 different clonal lines of B cells. And one of the things that you can see here too is that notice antibodies, the monomer itself, right? A single antibody in its monomer form has 2 antigen binding sites. Sometimes this is antigen binding to the, right, antigen binding site. So antibody binding antigen. Notice it's grabbing 2 at the same time. A lot of times this is referred to as the arms of the antibody, right? If we draw it as a stick figure, it's the arms. This portion sticking out is often referred to as the stem, but a better name is the FC portion. This is the fraction of the antibody that crystallized when they were studying it. But neutrophils, macrophages, right, phagocytic cells like them, have receptors for this portion of the antibody. Right? They're going to grab onto the stem that's sticking out. Do y'all see that? And then that'll help them gobble up this complex that we're looking at here. But we can also clump up antigens, right? Because look, we can bind to 2 of them at the same time. Right? So we can, one of the outcomes that antibodies can do is not only tagging for destruction that we're going to talk about, opsonization, as they can clump them up, or what's called agglutination. And this picture shows that. So what we're not going to cover again either, but as the name implies, it gives you an idea, right? Monoclonal antibodies, mono meaning what? 1, clone. Means 1 B cell, right? These are produced in a lab. Why are they so awesome? Because remember, 1 antibody binds to 1 epitope. So if we take this 1 B cell and we fuse it with a cancer cell, a hybridoma cell in a lab, we can get it to produce just this 1 antibody that we know binds to just this 1 epitope. So that's a very specific antibody, right? Why would we want to make those? Why would we want to make an antibody that only binds to one specific epitope? Yeah, because we know exactly what it's binding to, right? Right down to this molecular level of the epitope. So they're not cheap, although scientists have gotten good at making them, right? Hybridoma cells are immortal cancer cells which will grow in artificial culture, right? So, right, this is in a lab, right? We cause a mouse to produce B cells. We take out the B cells. We fuse them with myeloma cells, right? They become hybrid cells. And then we can isolate each B cell clone and isolate the single antibody that they're producing. So they're great for tests, right? ELISA stands for enzyme because there's an enzyme that we can link to antibodies. Immunosorbent assay. And those of you guys in lab, this is what we get to run the very last lab next week, as you'll actually run an indirect ELISA. A direct ELISA looks for the presence of a particular antigen in a patient sample. So notice there's antibodies, and as I said, right, these are monoclonal. They bind to a specific antigen. And then another set of monoclonal antibodies bind to it. But for these tests, they've designed a secondary antibody that will bind to primary antibodies. The difference here is it's got an enzyme linked to it. So when we add a substrate, a color change happens, right? This is how pregnancy tests work, y'all. Guess what the antigen is? Human chorionic growth hormone. What does that kit have? Why can we do these things? Because of the advent of monoclonal antibodies that bind specifically to antibodies. And this use of scientists tagging antibodies to recognize other antibodies. So we can flip this around too and do an indirect test where we have purified antigen and we can test to see if somebody's producing antibodies against it. But again, remember, these are proteins. This is all stuff you can't see. It isn't until if all of this is here and we add the substrate, which is not colored, right, and then it gets colored if the enzyme's there, then in plates like this, you can see the reactions. This is the control, right? But these are all different test samples that came up positive. Do you see the color? Where there's no color for the negative ones. Right? So they're pretty awesome, right? The ability to make these, right? But unless you have some problem, your body's going to produce lots of different. You're going to be polyclonal, right? Against it. So the general structure of an antibody is important. And we look at IgG, which is always in its monomer form. So, and this is your major class of antibody. So again, this is a stick figure of the poor antibody. They're proteins. They're made of two separate proteins, actually. Two identical separate proteins that you make two copies of. So notice chain A. Do you see this here? Right? So that's a protein, right? And it's actually globular, but we're mostly drawing in its stick figure form. And then this is B. See how much longer this one is? So guess what? Chain A, when they separated these antibodies, and notice you have one over here and one over here, it's a mirror image, is known as the light chain. Because guess what? It's smaller, so therefore it was lighter when they separated the pieces. The heavy chain, notice it's longer, so therefore it was heavier, right? So chain B in this picture is your heavy chain. But notice we produce two heavy chains. They bind to each other by sulfide bonds, because again, they're proteins, or remember, two amino acids out there. Methionine and cytosine have sulfur. So we get these sulfur bonds. The two heavy chains bind to each other, and they're identical. And then the heavy chain binds to the two light chains on either side. So it's a mirror image. Do y'all see that? So when you have IgM, right, they've taken 5 of these that are identical and hooked them all together to make that big antibody, that mega big antibody. So notice here too, right, the candy cane striping. This says variable region, right, because that's the part that's different for each antibody. And they illustrate this pretty well, right, as we looked, right, this part they drew it a little bit different. The solid color is what we call the constant regions. Constant because it stays just that, the same. For each class of antibody, this is going to stay the same because notice this is the region where they're hooking to each other. The two heavy chains are connecting to each other in that constant region. The two light chains are attached to the heavy chain. And then you have some intersulfide bonding within the chains themselves. But how we get this Y structure, right? Heavy chain to heavy chain, and then light chain to heavy chain, right, is because of this consistency. So IgG has 2 heavy chains and 2 light chains, as we saw as the picture, right? The disulfide bonds between the chains, inter, but intra within the chain itself. The good news, I won't make you distinguish between the difference of the two. As I said, typically most people just refer to those 2 parts that stick out, the top part right here, as the arms, because they were actually what grab on to the antigen. when they were studying antibodies and broke them up into the different fragments, that was called the AB fragment for antigen binding fragment. Where the bottom portion, sometimes referred to the stem, crystallized. And that's how it got its C. Not because they went ABC. That would be cool. It kind of works out that way, but so there's two ways we can break up an antibody. We can break these bonds between, right, and end up with 2 heavy chains and 2 light chains. Or scientists were able to break it in this hinge region. When they did that, they had this fraction and this fraction, which were the same, just mirror images of each other. And these contain the variable regions, which actually bind to the antigens. So again, they called that, right, the fraction that antigen binding happened. And there were 2 of them. And then they were left with one piece, this piece that sticks out usually when they're attached to an antigen, that crystallized. This is the part that phagocytes have receptors for. So this, we call this the FC, right, the fraction that crystallized, but there are phagocytic cells that have receptors for this portion. This is where the communication happens. Ooh. So, for antibody structure, remember we make B cells before we even see antigen. We'll make more once antigen's on board. We can make, they believe, anywhere from 1 million to 1 billion different antibodies. But you need DNA to make a protein, right? So, but we only have about 15,000 or so genes in humans. So how do we do this? Your B cells and your T cells, because this happens in your T cells to make T cell receptors, basically play the lottery all the time. They have 2 choices when it comes to making light chains. They have kappa and lambda. And for their variable region, there's 36 different variable regions that have been identified for kappa, and for lambda, another 36. So what your B cells actually do is they pick one of these segments, and they cut out the rest of the DNA. So like once they get programmed to make a particular antibody, that's it, they're stuck. Right? And then there's the joining segment. They have choices there for capta. And then they have that constant region, right, which is important so they can bind to the heavy chains. Little more choices for lambda. So again, they're going to choose one of these, right? One's on chromosome 2, one's on chromosome 22. It's kind of funny. Heavy chains is on chromosome 15. Again, they're longer. They still have a variable region. They have a diversity region and a joining region. And they have 5 different constant regions because they can switch between the, that's the part that determines what class of antibody we're looking at. Right? It's that bottom portion, that stem. So this next PowerPoint, which I ran all these together, didn't I? But that's okay. We'll just keep going. I won't separate them. Little bit of review, right? This chain, the smaller one, I don't know what the heck I clicked. Alright, it went away. Is what? Light chain. And B is heavy chain. This portion of the heavy chain is called the FC, right? And this is the part that's going to determine what class of antibody we're talking about, right? That's the fraction that crystallized. So there we got antigen binding. Here we have the one that binds to the phagocytes, the FC portion, right? We got diversity because they basically play the lottery. So here, right, the N portion, that constant region, this is what determines the different classes. So, what are the different classes? So, we make a whole bunch of this class. So this one is Ig, and I remember A because this is the most abundant one. And we actually secrete it. So look, it's two monomers hooked together by, guess what, a secretory protein. This one's called a dimer because it's two, right? Di for two. This is IgA. Remember, the most abundant, because this is the one that actually leaves your body and helps protect our surfaces. We produce this, it's in our mucus, it's in our tears, which is a different form of mucus. Women produce it in their breast milk. You produce it in your saliva, which is a mucousy substance. It's really good at binding. Look, it's got 4 antigen binding sites. It's really good at binding up antigen. Inside of our body, when it's not being secreted, it can bind to phagocytic cells. The next one, right, I always tell my students, this one is involved in allergic reactions, right? So usually my students will remember this one because I say, you know, just think it was named after me. E for Erica. IgE, right? I have horrible allergies, right? Most of the time when I'm stuffed up, right, I remind you guys I'm not sick. I'm not coming here to get you sick, right? My allergy medicine can't keep up with what I'm exposed to sometimes. What's unique about these is their Fc receptor binds to the surface of mast cells and basophils. I do not know why. I need to complain to them. They have eosinophils on this list. Cross it off. Right? And as I said, this only happens when it's inside of our body, right? Because phagocytic cells don't go outside of our body. Mm-hmm. Right, because then it's doing more agglutination and keeping it from sticking to us, which is something we'll talk about. Good question. Did y'all understand why he asked that? Right, and that's why I have this circled, right? So in this form, when it's outside of our body, this is not happening, right? And cross off eosinophils, because eosinophils don't interact with IgE. They interact with the most abundant inside of our body, and the special one that can cross the placenta, IgG, right? Always as a monomer. This guy, look, lots of abilities that we're going to talk about. But really important, right? This is the most abundant one in your blood. Serum is the liquid phase of your blood. And the fact that this one can cross the placenta. This is the one that causes problems with hemolytic disease of the newborn, right? Where Rh negative mothers, right, if they produce this antibody, can cross and attack the baby. The very first antibody, and as we found out, doesn't necessarily need T cell involvement for the B cell to make, is IgM. And I always remember mega big, right? Because this is the pentamer one. And this is present in your blood, right? And it does have some really important outcomes that we'll look at. I like pictures better. So, unlike T cells that really get locked into their T cell receptor, B cells, right, when they become activated, they can switch from IgM to one of these other classes with help from the B cell. We call that a course class switching. And they can mutate their DNA in hopes that the antibodies become more binding, right? Have more affinity to that. So this is a picture I stole from a different book to help us see this. So imagine you have a B cell, right? And it's binding to an antigen. You see that? And if we were to code it, we would say it's a 2+. It's pretty darn good affinity-wise at binding. So when these cells become activated and they go through clonal expansion, they actually actively mutate their DNA to the region that makes the antibody. So in this case, remember, mutation is random. In this case, the antibody they produced, notice the red color, not the strongest binding. So notice that it doesn't get stimulated to continue, right? This lineage stops, where the 2+ continues. So then it goes through another round of replication, right? Another round of mutation. And in this case, they kind of won the lottery because now they ended up with an antibody that had a much more greater affinity. This is a 3+. So notice that this line gets stimulated to continue. It has the best antibody, right? Has the best binding, the best affinity, where 2, right, he wasn't quite good enough. We'll quit making more of that. I think this picture does the best explanation of affinity maturation. So they literally mutate, right, as they go through each round of clonal expansion for B cells. The goal is that we get a better antibody because it binds better, right? More affinity, better binding. T cells are also going to help them make this switch. So remember, the first one they're going to make is IgM, always, right? That's the one that's on their surface. That's what they make and secrete. But with help of a B cell, they'll switch classes. So they'll, again, literally cut out the DNA and switch to making G. That lineage is stuck in that pathway, which what kind of sucks for those of us that experience allergy, right? Because for some allergens, we've gone the E route, and trying to get it to switch away from that is not easy. So what are our protective outcomes? Whew. I know we're cutting this close. So as the name implies, neutralization, right? What does that mean? It means when the antibody binds, we neutralize it. We stop it from being bad, right? Complement activation, remember complement, one of the things that can come out of complement, one of the outcomes we need to know is that MAC complex. What does that MAC complex do? It stands for membrane attack complex at that. It can cause what to cells that it can insert into the membranes? It can cause direct lysis and death of that cell. So some antibodies are really good at neutralization, right? Stopping the bad guy from doing its bad stuff. So we'll look at pictures of that. Some of them can activate complement, some antibodies, not all, and cause a direct lysis of the cells. Others just opsonize, tag it for destruction, right? So the phagocytic cells can come along and recognize it. Some antibodies are really good at agglutination, just clumping up the bad guys, right? Keeping them all in one spot. Or in the case of outside of our body, right, in our mucus, we clump them up. So again, our mucus moves this big clump of antigen antibody complexes out of the body or to destruction in the digestive tract. Some antibodies, right, are specific for activating eosinophils, basophils, mast cells. Remember, we also call these granulocytes, right? Because they're chocked full of vesicles that stained when we stain them. And they're given different names because their vesicles contain different substances. So they do different things, right? Eosinophils, right, release certain substances. We'll see in a moment. Basophils and mast cells are closely related. They have very similar substances, have similar reactions. And then you see this, right, acronym a lot. And I usually try to spell it out on the test. If I don't, ask me, right? I will say to you that ADCC stands for antibody-dependent, cell-mediated cytotoxicity. So when cells are coated in this particular type of antibody that can do this outcome, other cells come by and bind to it, but it's a specific cell and will literally cause the death of that cell that's coated in antibody. So let's see. Here's a virus-infected cell, right? It's probably expressing viral antigens on its membrane, hence why antibodies are binding to it. This cell, look, has an Fc receptor for the antibody. It binds to it, it recognizes it, it releases, right, perforin, granzyme, causes the death, programmed cell death of this virus-infected cell. What does NK stand for? Natural killer cell. So this outcome is that antibody dependent. Because look, the natural killer cell didn't know to kill this cell. It knew it because it was coated in antibody. Because remember, natural killer cells don't have receptors for antigens. Right? But they do have Fc receptor. So my next question is, there's only one class of antibody out of the 4 which we concentrate on, that actually can interact with the natural killer cell. Anyone know which IG this is? IgG. Think about, again, this is the most abundant one in your body. In your body, right? All right, so what do we see happening here? Here is, right, a bacteria. It's coated in antibodies. We've got a receptor here that's recognizing that portion of the antibody that we call the Fc portion, so we call this an Fc receptor. They say this is a macrophage or a neutrophil. So what are they going to do? They're going to eat and destroy this. But as far as the antibody, what job is it playing here? It's signaling the destruction, right, of this bacteria potentially, this antigen. So when we tag for destruction, that's called opsonization. Okay. So what outcome is this picture? So here we have a parasitic worm. Man, that sucker's huge. Are we engulfing this by phagocytosis? No. So instead, we have antibodies that are binding to it. And the unique thing about these antibodies that those of us that have allergens don't like is that when you produce this antibody, it attaches to this cell type. So it's primed. It's ready. It's what we call sensitized. So the minute the bad guy gets in, right, the antibodies are already bound to its surface. Do you see that? So this cell comes along, binds to the antigen, and immediately does what we call degranulation. Releases things like histamine. Notice what antibody this is. IgE. So anyone want to gander a guess of what cell this is? No. You would think it would be eosinophils, right? E for eosinophils. It's not. Basophils and mast cells, right? Eosinophil activation is not shown here because what antibody is needed to activate them? So, what class of antibody for basophils and mast cells, which we see in this picture? You see what in this picture? IgE. What class for eosinophils? Not pictured here. IgG. Yeah. So I know that one's kind of confusing. But if you can remember, right, mast cells, basophils, right, basophils, mast cells, bad, IgE, allergy. Erica hates all 3 of these. It'll usually help you remember. Am I out of time? All right. Is this the last one, though? No? Okay. We'll come back to these in our review on Tuesday. So remember, test is Thursday after Thanksgiving. Have a good Thanksgiving. I turned on, right, all the rest of the assignments were at the end. We only had one more, but that's okay. We'll go over them next time.